Right. All right. Well, it's 9:10. For everybody's here, welcome, to the biodiversity sessions here for the 2023 NISAC conference on the environment. I'm Steve Bisson. I've been involved with the EMC, the Tompkins County EMC for probably the last 10 years or so. And I'll be moderating this session. You'll be able to put any questions you have for the panelists into the chat, and they will be answered during the chat, the Q&A session after the four speakers have spoke. But you can put things in the chat anytime you want. You'll be muted the entire time here. You'll have to wait until the Q&A session for that. Also too, I just wanted to say, thank you for coming, and we really appreciate you, sharing your time with us. And for our first speaker, his name's Todd Bittner. He's the director of Natural Areas for Cornell Botanic Gardens. And his talk is capturing carbon, nature-based solutions to the climate crisis.
Session. If you hit X on your computer,
yeah,
it'll take you to the link.
Is everybody able to see my screen?
Yeah.
Great. Well, thank you very much. So here to talk about how we determined our carbon sequestration. Steve, I'm getting some.
Yeah.
You might need a mute.
All right.
Okay, thanks. Still getting some feedback.
Would all the panelists who are not speaking please mute themselves so we don't get any feedback? Thanks.
So Cornell Botanic Gardens is the Arboretum, Gardens, and Natural Areas for Cornell University. We can serve and manage more than 3,800 acres of diverse natural areas, representing all the natural community types and most of the rare plant habitats in the Central Finger Lakes region, and serve as the university's outdoor classrooms. Let me see if it's my speakers. Pardon me, I'm still getting some feedback.
If you check, if you're also on GatherTown, you want to mute that because you might get some cross feedback.
Okay.
Yeah, I don't know about you, John, if you're on GatherTown.
Can you hear it now? Okay, that fixed it. Thank you. Those same 3,800 acres of natural areas hold and sequester extensive amounts of carbon and are helping Cornell meet its ambitious goal of becoming carbon neutral campus by 2035. In the presentation today, we'll describe how we calculated how much carbon we are sequestering, how the data was obtained, and what the numbers mean. To determine the garden's annual carbon sequestration rate, we analyzed and mapped all plant community types across all of our holdings, following the classification system in ecological communities of New York State. We utilized multiple spatial data sets using ArcGIS that illuminate features in the landscape that influence what types of local plant communities can establish on a given area. On this map, you can see aerial imagery, topography, streams, and national, state, and local wetlands. Land use history, such as clearing for agriculture or timber harvest, can influence plant community types as well. Therefore, in this map, we layered in land ownership data, soil classifications, and added firsthand knowledge from expert staff at the Botanic Gardens to classify, age, and map the plant community types. What we found was a tapestry of 31 different plant community types, which represent the full range of ecological communities found in the Southern Finger Lakes region. The classification system we followed was from Edinger et al., Ecological Communities of New York State, 2nd edition, published by the New York Natural Heritage Program. Once we determined a baseline for our forests, meadows, and wetlands and gorges, We determined the carbon sequestration capacity for each. We then identified carbon dioxide sequestration rates per hectare for each vegetation type and 4 age classes for forests: 0 to 30 years, 30 to 70, 70 to 300, and over 300, as the age of the forest has a significant bearing on the sequestration rates. It's important to age the stands as well. The sequestration rates came from published literature, data from the U.S. Forest Service Inventory and Analysis Program, and data collected from Cornell University-owned forest lands. As not all of the vegetation types with carbon sequestration data directly matched the ecological communities of New York State, we needed to crosswalk between some of the vegetation types. For example, sedge meadows and old fields were both typed as grassland. Shrub swamp and successional shrublands were typed as shrublands, and rich mesic maple hardwood forest and successional forests were typed as northern hardwood, as just a few examples. At that point, we converted rates from hectares to acres, and using the community type acreages, totaled up the rates by vegetation type and by age category. We then summed all the totals and determined our total annual sequestration rate. Since many of us which was 10,685 metric tons of CO2 equivalent. Since many might not know and have a good frame of reference for what 10,685 tons of CO2 represents, we crosswalked it with Cornell University's carbon emissions inventory. As of 2019, the university's total baseline emissions for all of campus were 202,822 metric tons of CO2 equivalent. The garden's annual sequestration totals are equivalent to 5.2% of the university's total carbon emissions, or measured a different way, 40% of all faculty and staff vehicle commuting, or 43% of all university air travel emissions. But even modest-sized natural areas can sequester meaningful amounts of carbon. Here on campus, the natural area woods around Beatty Lake has rich soils and has been known historically for its high diversity of native and naturalized plants, abundant wildflowers, and rare species. The 48 acres of forest and wetlands around Beatty give us both beauty and carbon capture, sequestering 81.2 metric tons annually. The garden's Fisher Old Growth Forest is the best of the few remaining examples of pre-European settlement forest in the region. It is notable both for the extreme size of many individual trees, as well as 23 different tree species of canopy size. The 181-acre Fisher Old Growth Forest natural area, which also includes meadows and young forests, sequesters a staggering 437 metric tons of CO2 annually. Beyond our carbon inventory, an internal carbon sinks and sequestration working group is working to identify land-based approaches to sequester an additional 24,730 metric tons of CO2 per year. Afforestation and habitat restoration are among the primary approaches we plan to pursue to help take action to address the climate crisis using nature-based solutions. And so that's the end of my presentation, and I'm happy to take Q&A at the end of this session. You can see my email down below if folks are interested in learning more about our program or the literature where we got our carbon sequestration data from. I'm happy to share that with people. And with that, I will stop my share. Thank you.
Well, thank you, Todd. Yeah, the questions that you are proposing now in the chat, we will answer at the end of the sessions with the Q&A session at the end. But you can put in your questions now anytime you want. All right, for our next speaker, it's going to be Jeanne Grace. She's a Ithaca City Forester, and her talk will be about the importance of urban trees. Jeanne?
Okay. Are people seeing my screen?
Yeah.
Working? Okay. Okay, perfect. Okay, so yeah, so I'm Jeanne Grace. I'm the city forester. I've been the city forester for the city of Ithaca for about 13 years, 14 years. My email address is there if you want to jot it down. If you have any follow-up questions that we don't get to, feel free to send me an email. And I can put this up at the very end too. So today I'm going to be talking about the benefit of urban trees. So many people are going to be familiar with a lot of these, so I'll run through them kind of quickly. So urban trees definitely reduce the heat island effect. So the heat island effect is that, you know, solar radiation, that heat gets absorbed in the hardscape of cities. So paved areas, concrete buildings, things like that. And then that heat is retained and then given off in the evening when we're supposed to have cooler temperature. And it just creates this island of heat in urban areas that have a lot of hardscaping. And so trees create that shade and absorb some of that solar radiation. And so it mitigates that heat island effect. Stormwater mitigation is maybe something that people are a little less familiar with. So that functions in a couple ways. So when we have storm events, rain events, the rain comes, it hits the ground, it hits hardscape, that water is warmed up and then goes into storm drains and then right into creeks and waterways. And so along with bringing chemical pollutants or, you know, trash and things like that into the waterways, it also pollutes the waterways by increasing the temperature. So that warm water rushing into the storm drains and then into the creeks has ill effect for the biology of those systems. So what trees do is they have these large canopies, and in a light rain, they capture a lot of that moisture, and some of it doesn't even hit the ground. You think about being in under a tree in a light rain, and you may not even get wet at all. But even a heavier rain, it slows the rate that water is reaching the ground, and so it's giving the actual soil an opportunity to absorb that water rather than having it be runoff and just rush right into our waterways. So trees also clean the air, and so probably the first thing that people think of is the carbon sequestration, which Todd was talking about. So trees and other plants take carbon dioxide out of the air and convert that into sugar, and also just the actual structure of the plants as well. So it sequesters the carbon in that way. But it also particulate matter, so that tiny matter that we were all concerned about when we were having the wildfire smoke from Canada, that type of material also gets stuck in the nooks and crannies of trees and takes it out of the air as well. And then noise reduction, right? So you drive along the highway, you drive along Route 90 through, you know, the state here in New York State, and you see those like giant walls that are supposed to be sound reduction for the neighboring communities. Trees can also serve a similar purpose and are probably a lot nicer to look at than walls. And then the last one I want to touch about is a livable environment. And I feel like this is kind of underscored a lot of times, and we really focus on these other kind of more scientific things that people focus on, but just having a livable community is something that is really important to people, and it's a little bit less maybe tangible, but there are studies that have been done. People that are in hospitals have better health outcomes if they have even just a view of nature. We think about the Japanese concept of forest bathing and how refreshing and rejuvenating that is psychologically for people to have green space to be in, to just kind of de-stress, decompress, and feel better. And I think we've learned many things from the pandemic, but one of the things was how important green space and parks are for people. So when we have all this stress in life, to have a space to go out to relax, to exercise, is very important for people, and it's should be something that is seen as critical infrastructure. It's not an add-on. It's not something that's extra. It is something that is really important to the core of what living in a city is. So the thing to think about community forests and urban forestry is it doesn't just happen. It is a piece of infrastructure, and it does take effort. It takes planning, monitoring, maintenance, protection of existing trees, and community involvement to make sure that it remains a vibrant and functioning part of a urban infrastructure. So just an example to talk about is parking lots. So we reviewed our city code several years ago to improve some of our site plan requirements, and one of them was, so for when people are doing development projects, So one of them was about parking lots. And these are some images of some great representations of really poor parking lots and really poor environments for trees. So if you just are putting a tree in a parking lot on a design, that doesn't mean that tree is ever going to kind of reach that design intent of being a large mature tree that's going to shade that parking lot. Oftentimes in parking lots, there's very low soil volume and poor soil quality. And I'll use the term soil loosely because I've seen in incidents where the tree islands for parking lots are really just kind of construction rubble that's thrown in the tree pit, and then they put some mulch on top of it, and that's where the trees are planted. These islands are prone to have improper soil pH because of all the concrete and building materials that are around. That tends to have a more basic media for the tree to grow in, which is really not helpful and makes them lack certain nutrients that they need. And species selection is often not very good. So they're not picking species that can tolerate high temperatures, dry conditions, or kind of that, having super dry or super wet conditions back and forth. And so species selection is also not proper. And then establishment care, there's usually no establishment care. Trees can be planted at the wrong time of year. So they're being planted in like July and August when they're in full leaf, and they can't handle that stress of being transplanted at that time. And also they really need to be watered. It's very important for new trees to be watered while their root system is developing. So this is part of our ordinance. We had images like this that we had some students at Cornell through Nina Bassick helped us to create these images so that we could help developers to understand what we were looking for. So in this instance, this is showing a proper planting of trees in a parking lot. So here we have, this is actual organic soil that the tree would be planted in. Here's the tree island. And then out beyond the tree island where there's pavement, there's structural soil, which is a type of soil that can be compacted and you can pave over it, but it is loose enough and porous enough that tree roots can actually access it and use it. So this is looking down from above. So we have specs on, how many trees, how much soil volume each kind of tree would need. If you just have small crabapple trees versus a tree that's supposed to mature at a larger size, you might need to have structural soil on one side of it, versus if you have a tree that is supposed to mature at a very large size, you would need even more soil volume for that tree. So like I said, we require adequate soil volume. So you have to have a certain amount of volume depending on what size the mature tree is supposed to be. And we had a requirement that 25% of the parking lot had to be planting area, which is a quarter of the parking lot, which is like way more than any developer wants. But then we give incentives for upgrades. So if you want to, you have to start with 25% planting area. And if you don't want to do that, then you have to incorporate these either structural soil or these bioswales where water is funneled into the planting areas, things like that. Porous pavement is also a thing that is helpful too. So you can have asphalt that is actually porous, so water moves through it. And we have a couple experimental spots in the city where we have that, and the trees really do respond to that, and the root growth does occur underneath the area where we have structural soil and porous pavement over it. And stormwater swales is also really important. You can see these images here. So the idea of that is that when water hits the pavement, it is graded and funneled in a way that it goes into the actual planting areas. The important thing to remember there is you don't want these planting areas to become bathtubs. In a really heavy storm event, it can be a really significant amount of water that goes into these planting areas. And if there's no under drain or over drain, like kind of an overflow drain, those planting areas can just become bathtubs. And so you need to have, again, infrastructure. You need to have something so that water can drain out when it runs all the way through, and make sure there's an overflow drain as well so that we're not getting over flooding when there's excessive water going into these spaces. And when you think about a parking lot, where do people first park? Like closest to the store, but also in the shade on hot days. So, it makes the experience more livable for people as well. It's not just, it's not just about heat island and those other things that I mentioned. So what should you do in your community? You can encourage your community to view trees as critical infrastructure. It's not an extra. It's not an add-on. It's not something that's nice that we do just for aesthetic purposes. It is actually critical to have this as part of our urban living for us to have successful communities. You can review development ordinances in your community to see how trees are being protected, how development is being controlled so that we can protect trees and make sure that we're planting new trees, and use funding to purchase trees, but also create proper infrastructure for those new trees. There's a lot of availability for tree planting, and there's a lot of communities where the funding just to purchase the plant material is not necessarily the stumbling block that they need to get over. It is making sure that we have the infrastructure so the trees that go in do survive long-term and are successful. And then I do a lot of involvement to get the community involved, to speak with officials if they feel that trees are very important to them, share what they think the value of trees are. And I also involve the community in our work. So we put a lot of signs up on trees about when we're removing them, when we're replacing them, notices to people that live adjacent to ask them to water new trees, to have them be involved in choosing what kind of tree goes out in front of their house. So I find that to be very successful as well. So I will go back to my first slide. So anyone who wants to jot down my email address, you're welcome to do that and contact me if we can't get to your question. And I look forward to seeing the other presentations and answering questions in the Q&A. Thanks.
All right, thank you, Jeanne. All right, for our next speaker, Andrew Newhouse, the director of American Chestnut Research and Restoration Project at SUNY ESF, will give a talk about the transgenic American chestnut trees for environmental restoration. Andrew?
All right, thank you. Let me make sure you're seeing the right view here. Just a second. All right, is that the view you're expecting? Full slide?
Yeah.
Great. All right, well, thank you very much. I appreciate the chance to share today. Again, my name is Andy Newhouse. I work at SUNY College of Environmental Science and Forestry, or ESF, and I'm presenting work that some of which was done by me, but really we have a large and active research team involving several students, and so this is bigger than just my own work, of course. I'm going to talk about American chestnut trees, which are, a lot of people have kind of a historical concept of these trees, but I want to start with a little bit of background on what these trees were and how people used them and interacted with them. The nuts, of course, the chestnuts were a delicious food source for people, could be used in baking or eaten roasted directly. The lumber of chestnuts was straight grained and rot resistant, and there are some chestnut cabins and fences and things that are still in place today after more than 100 years. And so chestnuts have really worked themselves into our culture, into our society with street names and songs and so on. But maybe the biggest purpose that we're working toward at ESF is for environmental restoration. And some of the many animals that interacted with chestnut trees are shown here. They used the chestnuts themselves for food and the large trees for habitat and so on. So all of that changed in the early 1900s with the introduction of an invasive pathogen from Asia. The pathogen is a fungus that caused chestnut blight, and it enters through cracks in the bark or little branch scars, as you might see in this picture, and it usually results in the death of a tree within just a year or a couple of years after infection. But, and the chestnut blight pathogen kills trees primarily through this mechanism of releasing toxins. One of them is called oxalic acid, and I'm going to get back to why that's important in a little bit. But it doesn't, the fungus doesn't consume or doesn't rely on live tree tissue. It releases these toxins and kills the tissue, and then it eats the dead tissue, essentially. But chestnut trees are not extinct, and they're not even listed as endangered on most lists because they can resprout from stumps, and the chestnut blight fungus doesn't kill the roots of the tree. These sprouts can grow really quickly, but they don't typically reach maturity. They're not producing nuts commonly before they get knocked back by the blight again. So there are probably still at least millions of chestnuts on the landscape, but they're just not serving the same purpose and not serving the same function that they once did. And so we call them functionally extinct. There have been many efforts to restore or replant chestnut trees. One of the first ones was just planting Chinese chestnuts. The blight fungus came from Asia, and so there are a couple of species of chestnuts there that tolerate the blight fungus with almost no damage. And but those trees, like Chinese chestnuts, don't have the same traits. They didn't fill the same roles in our northeastern US forests that American chestnuts did. There have also been several other attempts that I don't really have time to get into now, involving various things like treating individual cankers and trying to induce mutations with radiation, and a more precise breeding program that has made some progress. But really, none of these have been successful in creating an American chestnut tree that has all of the traits people liked about American chestnuts and their roles in our eastern forests, and enough blight resistance to really be successful for a restoration tree. So our project at ESF decided to take a more precise approach starting in the early 1990s, using biotechnology to move a single gene. Instead of like traditional breeding that moves thousands of genes between related species, we moved one gene. This gene came from wheat. It's called oxalate oxidase. And you'll remember that the mechanism, the way the fungus attacks the tree, is to release these toxins under the bark. And this gene makes an enzyme, oxalate oxidase, that breaks down that toxin. It breaks down oxalic acid so that the tree doesn't get as much damage once it gets infected. And that's an important distinction, because this gene, this transgene, is not killing the fungus, it's not preventing infections, but it is allowing the trees to survive or tolerate infections. Just some other terms that we use. This means that the trees are transgenic, or we say GE for genetically engineered, or GMO. All of those are basically synonymous. And then the tree, the primary tree that we're working with that we've created is called Darling 58 or D58. So I'll also mention that both the transformation process, this idea of moving DNA between species, sounds really weird and unnatural to a lot of people, but this happens in nature. Even the same bacteria that we use to move DNA into the trees is a natural soil bacterium, and lots of trees already have DNA from this bacteria. So moving DNA across species and even across kingdoms happens in nature, and the oxalate oxidase gene is found in many other species as well. It's worth noting, I think, that the whole project here, that our whole effort to restore chestnut trees was initiated by members of the public. So the guy in the middle of this picture, his name is Herb Darling, and he's how we got the name for our Darling 58 chestnut. He owned a construction company in Western New York, and he found a chestnut tree in the 1980s and it got him excited about the history and the loss of the chestnuts. And so he approached these other two guys who were at the time professors at ESF, and that's what started the project. And so it's been important ever since for us to be able to communicate clearly what we're doing, what the importance is to a variety of types of people and audiences. So the transgenic trees themselves, like I said, still get blight, and the responses we see vary a little bit. The yellow circles on this slide are cankers or infections on transgenic trees. And so they're all pretty small, but you can see they're a little bit variable. In contrast, the non-transgenic controls that we've used often get infected so badly that the tree is snapping off at the infection site. And so it got hard to measure the cankers, for example, when we were doing this experiment last summer. But clearly the transgenic trees are seeing consistently less damage than the non-transgenic trees. The next logical question after, does this work, is it safe? And of course, that's a very important concern as we're hoping to do a restoration project. And I know you don't have time to read all of these, but I just want to point out that we've done a variety of types of tests to look at safety, to look at interactions with other organisms in the environment, and look at things like the nutrition of the chestnuts, and all really important points that in this case have all demonstrated that the transgenic chestnuts function the same way as non-transgenic chestnuts, except they can survive with blight. There are a number of ways that this project, that our restoration efforts are really unique compared to a lot of other projects. And one of them is that we want these trees to be really diverse. So this is really in contrast to agricultural products that you really want to be consistent, and you want the apple variety to be the same every year and on every plant and things like that. In our case, we're really looking to incorporate genetic diversity from as many different surviving chestnuts as possible. So, so we don't want something that's really consistent and genetically the same. We're trying to cross with, or we say outcross with, a number of different wild chestnuts to start incorporating diversity from around the range of chestnuts. And another important point is that the process we've used, the biotechnology tools, are tightly regulated. There are actually 3 different agencies that I don't have time to get into today, but 3 different federal agencies just in the US are regulating these. None of them have approved the Darling 58 so far, but we expect those approvals probably within the next year. So hopefully we'll be able to start distributing transgenic chestnuts pretty soon, but it will be a gradual process. And I need to unfortunately skip over a lot of this other interesting stuff, but just want to mention briefly that we're also looking at several other species of trees, or I guess I'd say we're starting to look at other species. There are chestnut relatives. A lot of people are familiar with American elm trees and of course Dutch elm disease, but there are other diseases on elms that would really be important to address before elms can be widely used for replanting. And beech leaf disease is another emerging disease that's really concerning and spreading quickly through New York right now. We're just at very early stages of starting to look at that. And then of course, a lot of people are familiar with emerald ash borer. We don't have transgenic ash trees yet, but we would like to start looking at other native hardwood trees. With that, I will share my email and take questions at the end of the session. So thank you very much for your attention, and I'll stop sharing now.
Yeah, thank you, Andy. All right, for our next speaker is Robin Bailey. And she's at the Cornell Lab of O, and her talk is about how citizens can engage and start up their own programs to support birds in their communities. Robin?
Great, thank you, Stephen. Can you just give me a thumbs up if you can see my screen? Did I share it correctly? Great. Okay, thanks everyone for the opportunity to talk about Nest Watch today and how citizen scientists can engage to support more birds in your own communities. So while citizen science may be a term that you've heard, it may seem like a relatively new term. It's important to note that people from all walks of life have been out there doing this kind of scientific volunteerism for many decades. They may have been called amateur naturalists or other terms in the past, but I think today we would call them citizen scientists or community scientists. And their work is really prevalent in the ornithology literature if you know how to look for it. In fact, the Cornell Lab of Ornithology initiated the North American Nest Record Card program back in 1965, making it one of the oldest citizen science programs at the lab. And people would send in data about birds' nests that they would find on these paper cards from all over the US for scientists to catalog and use. But sometimes they were already collecting this data for their own purposes as well, meaning they were already citizen scientists before we started asking them for these data. And these cards were amassed in a collection for decades at the Cornell Lab of Ornithology, where they served and still serve to inform natural history studies, particularly in the early days of ornithology. A lot of the North American species accounts were using this information for basic natural history. So in the late 1990s, with everything becoming more digital, this project evolved into an online program, and the mailing of paper cards stopped. It became known as the Birdhouse Network for a while, but eventually the name was changed to NestWatch. And NestWatch continues to this day as a long-term nest monitoring citizen science project with a rich history. And I currently serve as the project leader for NestWatch at the Cornell Lab of Ornithology. And sorry, I'm talking so fast. We only have 10 minutes, so I've got a lot to get through. hopefully it's being recorded so you can go back. So just a little bit of background before I start talking about how you can support birds. I'll tell you a little bit about how most people today are participating in NestWatch, and they're usually doing that individually. But there are a number of groups that monitor nests as part of NestWatch as a group, and they can range from organizations like nature centers, bird clubs, government agencies, community groups, and even corporations. And as you know, we've got this 58-year history of nesting data available to scientists, with more than 500,000 nest attempts recorded so far. That number is even out of date because it changes every day, but on average about 34,000 nests are submitted annually. And these have been used in a wide variety of scientific publications and resources. And what's nice about the protocol is that it can be widely used by many different kinds of people, from Alaska to Florida to Mexico, and the protocol is similar, so the data are comparable. And participants can monitor the areas and species that are of interest to them, and we help manage the data and provide useful online resources. And so I'm telling you all that because for this talk, I want to focus on how some of our community groups have been doing really great work that can serve as a model for how you or your agency, your company, your NGO, whatever it happens to be, can also engage and work to support more birds in your area, like some of the folks that I've had the pleasure of working with over the years. And I'll do that by featuring 2 case studies and then ending with some ideas that may be helpful for you. And the first project that I want to highlight is New York City Parks Wildlife Unit. As you probably can imagine, New York City is faced with a pest management problem, but also a public that loves any wildlife viewing opportunities. And they were able to come up with a unique approach to tackling a toxic problem. If you don't know, hawks, owls, kestrels, falcons, and other raptors do nest in New York City. You may have heard of pale male, the red-tailed hawk that nested there. It became famous, nesting in a fancy building, and people fall in love with these birds. But it became apparent that birds of prey are also vulnerable to what is called secondary rodenticide poisoning. And what that means is when city officials will put out rat poison to control rodent pests, another animal can eat it and then become either very ill or can die as a result of that poisoning. So what New York City Parks Wildlife Unit did was organize a group of willing volunteers to help combat this problem of raptor poisoning. And they did that by setting up a couple of goals for their project. They wanted to help city agencies make informed decisions about where and when to suspend the use of rodenticides. And at the same time, they also wanted to learn more about raptor breeding and nesting behavior in an urban environment. Since they're collecting that data anyway, might as well do some research about urban birds. And they've had a lot of great successful outcomes, including a comprehensive list of nesting raptors in all 5 boroughs of New York City. This has been, there's been ongoing monitoring since 2018 with the help of the community scientists. So they've got several years of data now, and upwards of 42 nests per year now being monitored for survival, which means that, somebody of the public or the parks agency is watching that nest and letting the city know that, you shouldn't use any rodenticides around this area and for this amount of time. So hopefully, let as many of those nests fledge as possible. So moving right along at a brisk pace, I'm going to talk about the next case study, which is based in Colorado, where some water pollution was affecting aquatic insect-eating birds. And this arose out of an environmental disaster known as the Gold King mine spill of 2015, which introduced 3 million gallons of toxic mine drainage into a tributary of the Animas River in Colorado. The very next year, concerned birdwatchers launched the American Dipper Project, which is a community science investigation into the potential impacts of this spill on breeding American dippers, which are an insect-eating aquatic songbird. Yes, it does swim. And it requires clean rivers for nesting. You'll only find it in clean, fast-flowing water. Unfortunately, to make matters worse, a wildfire in the area in 2018 caused flooding and introduced tremendous amounts of ash and sediment into the river, polluting it so badly that I was told 80% of fish died. So we've got this environmental tragedy. This group came in and set up some goals for themselves. They wanted to study the breeding success of American dippers on the Animas River and other streams in a post-pollution, I guess I'll say, environment. So they wanted to keep an eye on the birds and at the same time combat other threats to American dipper nesting success, such as wildfire, and possibly they're thinking now even microplastics. So they've also had some great outcomes. They've been monitoring aquatic macroinvertebrates with other partners in the area, which they have noticed have begun to recover. There was a bridge project that was scheduled to demolish a bridge in the area, which these birds can nest on the undersides of the bridge over the water. And because they were doing the monitoring, the participants were able to talk to the Department of Transportation and say, you know, these birds are nesting here, they're protected. Can you wait until after the nesting season to, you know, to do this new bridge project? And they were successful in that. So the birds were able to finish out their nesting season without being demolished, as it were. And so that's another great outcome. And they are monitoring about 54 nests per year for success and keeping an eye on things. So a great kind of community-built project that has helped to serve these birds in their area. Now, these projects arose on their own out of a local need. And I want to impress that the Cornell Lab did not set up these efforts, nor tell these people how to direct their energy or what to monitor. What we do is provide tools, resources, expertise when needed to help further the goals of community engagement. But what I want to emphasize is that if you see a need in your community and you have an idea for how to respond to it, I want you to feel empowered that you can do that. You don't need to be an ornithology expert. And one of the tools you can use, I'll just quickly remind everyone that the Migratory Bird Treaty Act makes it illegal to take or kill birds, their nests, their eggs, or any of their parts. So if you notice anything happening in your community, whether it's the spraying of poisons or the removal of birds' nests because they're in an inconvenient place, and that results in the death of birds or the loss of a nest, you can take action, and you can use this law as your tool that you can pull out and say, hey, building manager, I noticed you were taking down bird nests off the building. Actually they're legally protected. And this does include unintentional take or accidental nest removal. So, ignorance or accidental is not an excuse. Okay, so I told you all that, but I also want to give you some ideas for community projects that can be undertaken when there is no environmental disaster like a toxic spill or poisoned birds that you need to react to. Sometimes we can just proactively help birds, and that's awesome too. Here are some examples of things that my husband and I have helped organize in our small community of Lansing, New York. We've worked with the community to put up nest box trails, particularly in public natural areas and walking trails. Hopefully targeting some declining species in our area, but also bringing the public into contact with more nature and allowing them to see nesting birds at probably a closer range than they otherwise might. We've also worked on nesting platforms for ospreys. Ospreys have really made a comeback in the Finger Lakes and like to use utility poles to nest, which is unfortunately a hazard for both them and the utility company. So, we've been working with NYSEG in our case to put up these artificial risers on poles that are not related at all to the power. They're just disconnected and placed elsewhere. And so this is a great project that maybe is something that could be duplicated anywhere where there's lakes or bodies of water. We've also worked on chimney swift nesting towers, mostly with an Eagle Scout who did that project. So this is a great Eagle Scout project, but providing some guidance and some labor to help make that a reality. Chimney swifts are a bird that nest in people's chimneys, and understandably, some people want to close off their chimney so the birds can't get in there. But you can make a sort of freestanding chimney for them that the birds can nest in and put it at a nature center or somewhere where people do want birds nesting in this structure. And another idea was just maintaining grassland areas for ground nesting birds. So many birds do nest on the ground, which surprises a lot of people. And you can work with your community to delay mowing or come up with a managed mowing schedule so that the whole field isn't being mowed all at once, ideally after August. But these are just some ideas. The possibilities are really endless and can be adapted to many different types of communities and the interests of the people that live there. So with that, I think my time is up, maybe over. So thanks for your attention. And there's my email.
All right, well, thank you, Robin. All right, so now we're at the question and answer time for any of the panelists you have. John Rhodes is going to moderate the chat, and so if you have any questions, please put them in the chat for our participants here. And okay, John, why don't you go ahead then and tell us our first question.
Since we just finished Robin's presentation, there are two questions for Robin. One is, could you provide resources about delayed or managed mowing in grassland area for ground nesting birds?
Yes, that would probably be easier for me to do via email. So if you want to email nestwatch@cornell.edu, I can send you some resources for that.
Great. And the second question for Robin, is it illegal to remove nests from residential properties?
It's illegal to remove any nest that is active, meaning it has eggs, young, or the birds are actively building it. If it's an old nest from last year, you can take it down.
Okay, and we have a question for Todd. You used the term afforestation in your presentation. Could you please define that for us?
Sure. Typically, it is areas that have been converted. They're no longer forest, agricultural fields, old fields, those kinds of habitat types, and intentionally reforesting them by planting trees. So afforestation is creating forests where there weren't any previously.
And a question for Andrew that I'm not 100% clear about, but it basically says, can you treat Darling 58 cankers that arise with oxalic acid directly?
That would probably be using oxalate oxidase to use the enzyme directly instead of as a transgene. And that could potentially work, but the interaction between the fungus and the tree is taking place in inner layers of bark that are not very accessible. So that would probably be difficult, like a topical spray probably wouldn't be very effective. So you'd be looking at some sort of injection, which is possible. That is done with other types of trees. But probably simpler than that is a technique called mud packing. And if you just scrape off the outer bark and squish some mud onto the infected site and wrap it in plastic so it stays moist for a few months, that actually treats that individual canker. It doesn't heal the rest of the tree and it doesn't prevent future infections, but it's a good way of keeping trees alive as kind of a one at a time basis.
Okay, and yes, we have another question. Is there research regarding trees or critical infrastructure? That's a quote, trees are critical infrastructure. And can you send that out? I think that would be very helpful.
Yeah, I don't know. I think that's just something I say that, that's how I try to like approach like our common council and our public works, is to just include trees as infrastructure, not as an add-on idea. I don't think there's that I know of like a research project that shows that. It's just kind of the concept of how to get a community to think about trees and treat them as an important factor, as important as making sure the stormwater runoff system works, or that the roads are paved, or that the streetlights are running. Having trees is also a part of infrastructure that communities need to focus on.
Thank you. I think that's all the questions that we have, Steve, so far.
You're muted, Steve.
Steve, can you unmute yourself?
Yeah, sorry. Okay, good. I'm glad we're right on time. And so for our next speaker is Jeremy Hurst, and he's with the New York State DEC Division of Fish and Wildlife, and his talk is about New York deer management. Jeremy? I don't know if John, if you made Jeremy Hurst.
Oh, I'm sorry. Is he not? I apologize. Just a second. Let me find him.
Sure.
There we go. Okay.
And Jeremy, are you unmuted?
I am now. Are you seeing my screen or just me?
Yes, no, I see your screen.
Perfect. All right, so let me make sure I got it set up properly here. Okay, well, thank you, everyone. Good morning. As Steve indicated, my name is Jeremy Hurst. I work for the Department of Environmental Conservation within our game management section, overseeing our deer program and other game management programs for the state. And so I want to give you a kind of a high-level view of deer management in New York, as well as bring it down to the small scale that many of the communities that you all are engaged with may be interested to learn about. And so I've subtitled my talk an ecological and social imperative. And I think it's an important perspective for us all to consider as we think about deer, which are a beautiful, graceful species, important for New York, valued by many New Yorkers, but also very ready to create some problems. And so the two images on your screen now, on the left, you see a forest that's been partly fenced to allow native regeneration to occur, absent any deer browse, and then the typical forest to the side that has been experiencing excessive deer browse. These are really common experiences in suburban and urban portions of the state where deer populations have been allowed to increase without sufficient mortality, and the browse level from deer can dramatically change the ecology of that system. And on the right, of course, we see deer in the suburban yard, where we know deer can create havoc for landscape plantings and gardens, risks for deer vehicle collisions, as well as providing a host for ticks, which can carry many different diseases. And so these are concerns both from a social factor and ecological factor, something that we look at a statewide level, but also try to work with communities to resolve at the local level. And so our state management plan is available for anyone to review. It covers all aspects of deer management, both from a habitat management, population management, wildlife conflict damage management, hunting regulated activities. All aspects of deer management are covered in here. And so I won't dwell on this in detail, but it's available for your review. As for how we manage deer in the state, hunting is the primary tool. And so we've broken the state down into 92 management units, 89 of which are lawfully allowed to be hunted. And we adjust deer populations in those areas through a series of regulations. As you can see on the map on the right, deer hunting is highly regulated. All hunters in the state have to go through a hunter education course to be trained in order to do so safely. And we regulate the deer populations throughout the state using hunters and varying both the seasons as well as the tags that we make available to hunters to harvest deer. The management process starts with public input and an assessment of forest impacts. We want to know what the social and ecological impacts of deer are in particular areas, and it helps guide our management direction. Currently in the state, almost the entirety of the state is in a status of wanting to maintain deer populations at the current levels, though there are areas in central New York and southeastern New York where we want to reduce deer populations. In no part of the state has our guidance been given to increase deer populations. However, that's occurring naturally in some areas where we have limited control over deer, particularly in urban and suburban areas. But as you see, once we set our management direction, We use hunting as the primary tool to adjust those deer populations to meet that management direction. We assess the population, we adjust our tags and seasons in order to accomplish the necessary harvest, allow the hunting season to occur, and then readdress. Over time, we continue to reassess forest impacts and reassess public values for deer and their impacts in order to modify the management direction. But because deer impact people at a variety of levels, so do our management programs. And so at the state landscape scale, I showed that image of our season maps. We regulate deer hunting intensely by adjusting the number of deer management permits. This is the DMPs there at the WMU scale. The deer management permit is an antlerless tag, and we set quotas for all of our management units as to how many antlerless deer we want to remove from each unit in order to achieve the desired population change. We can also adjust the bow hunting and muzzleloader seasons in order to, again, influence some degree of antlerless harvest. And we can create extended seasons if we need to in some areas. But as we get to smaller scales, the town, community, property level, we have to be a little bit more intensive. And so we work frequently with communities across the state in order to establish more controlled hunts, allow them to use regulated hunting in the typical framework, but in a more refined fashion, how they can work with hunters in
Anyone
their
who's
community.
in the yellow room and can hear me, if you want to join the biodiversity session, which is in progress, please click X on your computer, and that will take you to the Zoom link for that panel discussion. Just click X on your computer, and it'll take you to the Zoom invitation for that session. Thanks a lot.
Right, and I'll continue, and you can see on the lower right, the more intensive programs are our Deer Management Assistance Program and a Deer Damage Permit Program. The Deer Management Assistance Program is a hunting-related program, uses our regulated hunting seasons, but it provides more tags for property owners or a municipality in order for them to harvest additional antlerless deer. You may also be familiar with terms like a deer cull, a sharpshooting program. Those happen through deer damage permits, where we allow municipalities or landowners to remove deer outside the typical hunting framework, and with additional tools like using bait or shooting deer at night with lights, more effective, more efficient, and more specialized. And so we work with communities to develop those programs as needed. We have a whole suite of information on our website. A lot of work has been done in this in New York and nationally on how to help communities be more effective. And so to the left, you see our community deer management guide that's available for your review on our website, as well as a suite of other materials that we've worked with other entities to develop over the years. This can all be found on our webpage related to deer overabundance. But I'd also want to direct you to a program that was developed by Cornell, the Nature Conservancy, and we helped to consult with. And this is the Community Deer Advisor website. There are great examples of other communities that have been working through deer management issues, a suite of resources, maps of other communities across the country. And an important consideration here is this issue evolution of how deer management works in communities. And we see a typical pattern, community to community, where we need to define the problem, work with the community to help assemble public within that community to make some decisions about the path forward that's right for the community. We can help guide and provide some technical advice as to the best way to implement various programs. And then, of course, we always encourage communities to set up a program for evaluating and monitoring the impacts of deer to determine whether or not they're actually being successful in addressing the deer population. But deer management at the municipality level needs to be comprehensive, integrated, and progressive. It needs to involve both non-lethal and lethal approaches to address the deer population. It needs to integrate not just big green spaces, but small green spaces. And it needs to be progressive in increasing the escalation of impact in order to achieve deer management. And I'll say that if your community is not already involved in deer management, you will need to be at some point. And so the communities that we see most effectively dealing with deer management address it early and proactively. They start with low-level investment, passive management, allowing regulated hunting to occur in spaces where it could legally by state law, in addressing municipal discharge ordinances and removing the local laws that prohibit discharge of firearms or archery equipment. And that just allows hunters to do their thing in the green spaces that are occurring within a municipality as would otherwise be allowed by law. That type of low-level investment early on can prevent a deer population from growing. But unfortunately, what we see more often is that communities neglect that and wait until there's an escalated problem and deer are chronically a problem in their community, creating all sorts of issues to levels that become political, and then becoming or integrating a deer management program can be much more difficult. So start by documenting the impacts and working to generate community support, looking at non-lethal approaches to reduce deer impacts, maybe enforcing a feeding ban, identifying areas within the community where deer are most active, and working to address deer populations in those areas. Communities can implement controlled hunts, like I mentioned. They can augment with deer damage permits, such as sharpshooting. And then even in the deer management plan, as I noted earlier, we have an appendix that identifies a variety of statutes, state laws that are actually impeding local communities from being successful. So you can advocate for legislative change that enhance our tools. And then in some cases, surgical sterilization as an option of reducing a deer population in a really urban core may be an effective contribution to an overall program. As I indicated, monitoring impacts and success is important. You'll hear later this morning from Christy Sullivan, actually I think next, about the assessing vegetation impacts from deer, a project we worked on closely with Cornell Cooperative Extension. But communities can also monitor landscape vegetation within their boundaries, and how planted plants or potted plants may be impacted by deer as a means to monitor impact. And keeping track of deer vehicle collisions and changes therein can be an important tool for monitoring success. So here's my email address and our web link. Feel free to reach out to me if you have additional questions, and I'll stick around for the Q&A later. Thanks for your time.
Thank you, Jeremy. All right, let's see. All right. Now, just for any new people who are here, we'll have questions and answers at the end of the 4 speakers sessions, and if you can put them in the chat, and we'll answer them at the end. All right, for our next speaker is Christy Sullivan. She's with the Cornell Department of Natural Resources and Environment, and her talk will be about the AVID assessment method as a tool to measure the effect of a deer on forest biodiversity in the community. Christy?
Okay. Can you all see my screen?
Yeah.
Is it on regular presenter mode, or is
No.
no? Can fix that.
I mean, it still was pretty big, so
It was pretty big.
I could read it.
Okay. Oops.
Much better.
Yeah.
Better?
Yeah.
You can see it?
Yeah. I think if you just do F5, isn't it? Then that.
Yeah, I tried to do F5 and then it
oh,
puts it
didn't work.
into
Yeah.
a weird mode. Doesn't usually do this. Hang on.
I think that's probably good enough.
Yeah.
Okay. If it allows me to. Okay. Okay, good morning, everyone, and thanks for the lead-in, Jeremy. My name is Christy Sullivan. I work at the the Department of Natural Resources and the Environment at Cornell University. I'm a wildlife biologist there and an extension associate, so I do a lot of outreach on a lot of a variety of different wildlife-related issues. Today, I'm going to talk about AVID as a tool to measure the effects of deer on forest biodiversity in your community or on your lands. I'm going to start with an overview of what those deer impacts are and what they look like, and then move into the actual nuts and bolts of the AVID methodology. So a variety of things make a healthy forest, but one very key component is that a healthy forest can have a variety of native plants and tree species, and that the forest has the ability to grow or regenerate in the case of disturbance. And that could be a natural disturbance or a timber harvest, which is intentional. But basically, having young trees and shrubs that can take over if the forest canopy is lost. So in DEC's Deer Management Plan, the most recent plan, they mention a term called regeneration debt, which was coined by Dave McGill in a paper in 2019. And that's basically regeneration debt is when you have a canopy of trees, a full, nice canopy, maybe a variety of species or maybe just a few species, but that there aren't enough seedlings in the understory that if those canopy trees are lost, that you would have a new forest to grow up and replace the one that's currently existing. Either you wouldn't have a forest at all, or you would have a forest of a much different species composition than what you currently have. So DEC shows in the Deer Management Plan this regeneration debt map for New York State, which was, I believe, a modeling effort with SUNY ESF. And basically the green area is where there's some decent chance of having regeneration, that less than 25% of that area is experiencing moderate to severe regeneration debt. The tan area, which covers the majority of the state, is where you would have over 50% of the forest with regeneration debt. And then not acceptable is where more than a quarter of the forest in those blue areas are experiencing moderate to severe debt. So just to give you a picture, a very vague picture or overview of, or not vague, but very like a simple snapshot of regeneration debt in New York State. So deer are unique in that they have the ability to change their own habitat and the habitat of other species. And they do so in a number of ways, primarily by what they feed on, how much they eat. Deer need about 8 pounds of food per day per deer. So when you think of what would constitute 8 pounds of seedlings on the forest floor, that's a lot just to support one deer. And so not only do they eat quite a bit, but they also are very selective in what they eat. And by choosing some things over others, they are kind of pushing the ecological community toward those species that they don't like, so they can change the composition as well. So one way that deer affect the forest is that they change either the forest structure or the plant species that are present. And Jeremy showed a similar picture of an exclosure, but in this image to the left is the area where deer are still kind of wandering about, and on the right is an area where deer are excluded. You see the fence there dividing them. Otherwise, that stand was treated in the exact same way on the left and the right. The same number of trees were removed, but you see that on the left, you've lost that structure. You don't have a nice understory coming up, and the diversity is very, very low. You have basically a stand where ferns have come in, taken over the understory, and when you have that condition, it's very hard to reverse because ferns also prevent seedlings from becoming established. So in addition to tree species, deer also affect shrub and wildlife species, wildflower species richness. In northwest Pennsylvania in Allegheny National Forest, they did a study in what is considered the oldest deer exclosure. It's about 60 years old. And they showed that even, all these years later, the wildflower and shrub diversity was far, far less outside of the exclosure than within the exclosure. And they concluded that overall, deer browsing in the region has caused about a 60 to 80% decline in the number of wildflower and shrub species regionally. And also many of these wildflowers have very low dispersal and reproduction rates. So it'll take many, many, many years for those species to be able to rebound, even in the absence of deer browsing. Another way they affect the forest is that by removing native vegetation and browsing it, it creates kind of a disturbed condition, an opening per se, where it gives kind of invasive plants the opportunity to move in and kind of take over. They also affect the forest by reducing or eliminating habitat for other wildlife species, and birds are often mentioned. There are many, many papers out there describing the impacts of deer browsing to bird habitat. And I just use this one by Nettle et al. To kind of highlight it, but they found that there's a legacy effect on bird density. So where there are higher densities of deer during forest stand initiation, so basically when those seedlings start to grow up or they're trying to regenerate a new stand, because of browsing and removing vegetation, there's a lower canopy foliage density, so fewer leaves. And as a result of the fewer leaves, there's also fewer insects for birds to feed on, and that leads to bird density, decrease in bird density. The other issue is that a lot of birds breed or nest in either the shrub layer or the understory layer. And so when those layers are missing, all those species that would nest there have no place to nest. And it has been shown that there's been quite a decline in most or many of the species that nest in those layers of the forest. So what is AVID? AVID is Assessing Vegetation Impacts from Deer. It's a rapid assessment method for evaluating deer impacts to forest vegetation. It's a method that we developed here at Cornell in cooperation with Jeremy and funded by DEC. So who can use AVID? Well, pretty much anybody can use it. There are some school groups that are using it, but also landowners, local governments or local government organizations, community groups, hunters, foresters, land managers, land trust personnel, and volunteers. And it's basically a method where you collect information about tree or shrub seedlings on your land or lands in your community, lands of your clients if you're a forester. And some of the benefits of using AVID is that you get to learn about the ecology of the land, learn to identify tree seedlings. You don't have to identify too many, just one or two that you plan to monitor. You get to develop an eye for recognizing key signs of deer impacts to your forest, and to help be part of an effort to document the health of New York forests and track changes over time. So you contribute to the statewide effort to document deer impacts at a more local scale than we would have otherwise. Just kind of in a nutshell, AVID, to participate in AVID, you go out and you establish 5 to 6 plots of 6-foot radius and kind of mark a center stake, put in 4 corner flags or 4 flags in each cardinal direction, and then you find and tag either 5 to 8 tree seedlings in each plot and measure them once a year for 3 years. So 4 measurements total. The first year when you establish the plots, and then you just go back and measure 3 more times once a year in approximately the same time of year that you establish the plots to begin with. So the most effort is setting them up to begin with, and then when you revisit, it's actually quite easy in the years following. You can collect data on paper data sheets that we provide on the website. And if you choose to do that, you can enter the data on the website. So there's a portal where you sign up to use the website, and then you can enter the data. You can also visualize those data. It'll provide graphs showing you your seedling data over time. So that's a nice function. And there's also a lot of good information on the website, the manual that gives you the overview and the details of the methodology, and some other information as well. You can also, if you don't like to use paper, or actually probably an easier way, more streamlined way to collect the data is to use our AVID app. It works quite well for either Android or iPhones. You can find the app at Google Play Store or the Apple Store. It's called AVID Deer. So AVID, when we developed the method, we tested it ourselves, using AVID to compare deer browsing or the effects on seedlings inside and outside paired exclosures across the state, and monitored those for 4 years. And then we're able to show that yes, AVID is an effective tool for showing impacts over about three growing seasons for the most part. And as AVID data become available, they'll be available to DEC to help inform decision making. So the key is getting people involved. So the more people that participate, the more data we have on the ground, and the more information DEC will have. If you have any questions, or if you would like seedling tags, I have quite a few thousands of pre-numbered seedling tags that I'm happy to send to anybody who wants to participate. Feel free to visit the website or contact me anytime for more information.
Great. Thank you, Christy.
Mhm.
All right. All right. For our next speaker is Caitlin Deutsch. She's with the Cornell Entomology. She's a PhD student, and her talk will be about sustainable beekeeping. Caitlin?
Great. I'm excited to be here. I'm going to switch over to my presentation. Can you see this in the presentation mode? Great.
Yep, looks good.
So yes, thank you. I'm excited to talk to you all today about sustainable beekeeping. I am a PhD student in the McArt Lab at Cornell University, and my research is really focused on how honeybees can share diseases with wild bees. So today I'm going to talk through how wild bees and honeybees can interact, a little bit about my research that shows some problems, and then I'm going to zoom back out and look at potential solutions that are ongoing. So the question I'm going to keep coming back to is, can we improve honeybee keeping practices to not only support healthier honeybees, but also wild pollinator biodiversity and health? So to do that, I'd like to start here. I'm sure many of you have seen a lot of these Save the Bees campaigns to raise awareness and help reverse pollinator declines and losses. And it's true, both managed honeybees and wild pollinators are facing really serious population declines. However, one of the commonly offered solutions to these declines are adopting a honeybee hive or taking up backyard beekeeping. And so I would like to share how this can actually have some unintended negative consequences on wild pollinators and offer some potential solutions. So when I'm talking about honeybees, I'm talking about the European honeybee. The species name is Apis mellifera. As the name suggests, this is not native to North America. It was brought here from Europe, and it's managed as a domestic species. So we can really think about honeybees as the livestock of the bee world. They provide these critical pollination services as well as honey production. But managing their population declines is not an issue of biodiversity conservation. It's truly an agricultural security issue. However, there are also a number of incredibly diverse pollinators in New York State. We have over 400 species here in New York, and these also are capable of providing pollination services into regionally important crops like apples and strawberries and squash. And unfortunately, they're under the same stressors as honeybees. So a recent report, the Empire State Pollinator Survey, showed that almost 40% of insect pollinator species are at risk of being lost from the state. So why are these declines happening? There are a number of stressors, including pesticides, climate change, habitat loss. But today I'm just going to focus on pests and pathogens, so disease pressures that both managed honeybees and wild bees share. So in the era of COVID and the pandemic, I don't think I need to impress upon anyone how devastating RNA viruses can be. The main player in honeybee colonies is deformed wing virus. This can lead to a lot of colony death, and it can be shared between bees when they share food resources, but it's also vectored by this really nasty mite that you see here. This is called the varroa mite. It's basically like a big tick for bees. So not only does it feed on bee tissues, weaken the bees, but it can also inject viruses directly into bee tissues and bypass a lot of the defenses that bees have. So this problem looks like this. When you have higher varroa mites in your honeybee colonies, those bees are often sicker. They have elevated levels of deformed wing virus. And so beekeepers are constantly having to monitor and control for varroa mites to keep their bees healthy. What does this have to do with wild pollinators? So varroa mites are specialists on honeybees. They cannot parasitize and feed on any other bee species. However, those viruses that they transmit can be shared with wild pollinators and cause infections and death. And there's this growing concern that these viruses are spilling over from honeybee colonies and into wild pollinators across shared floral resources. So that happens when an infected honeybee visits a flower patch around the apiary. While she's feeding, she may defecate and leave behind some viruses. And then the next wild bee that visits that flower patch can be exposed to the virus, and if it's susceptible, will develop an infection. So the question that my research is asking is whether controlling varroa mites in the honeybee colony can actually reduce transmission of the virus from honeybees to wild pollinators. So essentially, are varroa mites actually the drivers of this pathogen spillover of deformed wing virus? And if so, what are potential solutions to mitigating that spillover? So as part of my dissertation research, we went to 27 apiaries all around New York State. And at each site, we measured the varroa infestation load. We're measuring this as varroa levels per 100 bees. We also measured the amount of virus in those honeybees. And then we sampled goldenrod flowers and bumblebees in order to see if the virus was there. So we verified that yes, when there were higher varroa levels, those honeybees had higher virus levels. And so then I'll explain what effect this had on flowers in the apiary. So this figure is just going to show here on the x-axis the average varroa load per 100 bees. Oops, sorry, my screen went out. Can people still see?
Yeah, I still see everything fine.
Yeah, I see
Okay,
the presentation fine.
Thank you. Sorry about that. Yes, so on the x-axis here, it's average varroa load. So how bad are varroa mites at each apiary? And then here on the y-axis is the prevalence of deformed wing virus on goldenrod flowers. So the proportion of flowers at each site that are testing positive for the flowers. And we see that the higher the varroa levels at each apiary, the more flowers at each site are testing positive for the virus, suggesting that honeybees are getting sicker and they're depositing more virus on these flowers. And so is that having, we need to now figure out if that's having an effect on the deformed wing virus in wild bees. So again, this is the same x-axis here, how much varroa is at each site. And now on the y-axis is the prevalence of deformed wing virus in bumblebees and wild bees. And we see the same trend where the more varroa mites are at an apiary, the more bumblebees have deformed wing virus as well. And this is so incredibly striking. The site that had the highest varroa loads, every single bumblebee we tested was positive for this virus. So to kind of summarize all of this research, varroa mites lead to elevated levels of deformed wing virus in honeybee colonies, which then leads to more virus being deposited on flowers surrounding the apiary, which ultimately leads to more bumblebees and wild pollinators being able to pick up and be infected by this virus. However, I don't think this is all bad news. If we come back to my question of can we keep healthier honeybees to support wild pollinator diversity and health, the answer is yes. I think this shows a real opportunity that beekeepers can have this key role in not just protecting the health of their honeybees, but also the entire wild pollinator community, simply by adopting more sustainable beekeeping practices. So what can they do? The first is to improve monitoring and controlling varroa mites in colonies. This is really difficult, but here at Cornell, the New York State Beekeeper Tech Team has already begun partnering with beekeepers to give them knowledge and tools to implement the best management practices for varroa mites. And this has been super effective. So after enrolling in the program, beekeepers have reported to increase monitoring efforts in their treatments for varroa mites and have halved the number of colonies lost to varroa infestation every year. Another consideration for beekeepers is to avoid placing apiaries in sensitive habitats that might host imperiled pollinator species. So this is another graphic from that Empire State Native Pollinator Survey. The dark green are protected areas that have conservation mandates. So I like the idea of using these maps as a guide to where you can minimize the effect of placing honeybees. And so you might want to avoid putting honeybees in very sensitive habitats. And then finally, whenever possible, plant flowers and advocate for more flower plantings. When bees have access to a more diverse and nutritious diet, they're better able to fight off disease. So in conclusion, honeybee health, wild bee health are tightly linked. I hope I've convinced you that adopting honeybee colonies is not the most effective pollinator conservation measure, but we do need both. We do need to have both honeybees and wild bees. So making sure that we're keeping our honeybees in a sustainable way will help mitigate any negative impacts on the wild pollinator community. Lots of people I would like to thank, but I'll end it here. I'm happy to take any questions, or you can email me, or I'll be around at the Q&A. So I'll pause here.
Thank you, Caitlin. All right, for our next speaker, is Kristen Schuler, and she's a wildlife disease ecologist at the Cornell Wildlife Health Lab, and her talk will be about the New York State Wildlife Health Program.
Great.
Kristen?
Can you see the slide appropriately?
No.
Okay.
Are you sharing your screen?
Yes, I did share my screen. Let's try that again.
If you have any troubles, I can always try to do it on my end too.
Yeah, let's hit share. Do you see it now?
Yes.
Okay. And is that correct?
Yes. Yep.
Okay. Excellent. Well, thank you for having me here today. It's really a pleasure for me to introduce this program. It's the New York State Cooperative Wildlife Health Program, which is a partnership that started in 2011 between New York State's Department of Environmental Conservation and Cornell University's College of Veterinary Medicine. And our work supports DEC's mission to safeguard the long-term health of wildlife populations in New York. And in accordance with One Health principles, we partner with human and domestic animal health experts in situations of mutual concern. So we have core staff that are based at the Wildlife Resources Center in Del Mar, New York, and at the Cornell Wildlife Health Lab, which is based at the Animal Health Diagnostic Center at Cornell University. The goal of our program is to promote the health and sustainability of wildlife populations through the integration of wildlife ecology and veterinary medicine. We have 9 major program areas within the wildlife health sphere. Those are surveillance and monitoring, preparedness and response, training and development, communication, information management, research, veterinary health services, policy development, and then program management. So I'll just highlight a few of these today. The biggest one and the one that we spend the majority of our time on is health and disease surveillance, because this improved disease surveillance helps us establish disease patterns and also more rapidly detect and respond to new and emerging threats that can impact wildlife and human domestic animal health. So we share relevant case data with our counterparts in other state agencies to alert them to disease issues of consequence. So what typically happens is that a person may find a dead animal and they call the DEC to report it. And if that falls within our criteria for examination, the DEC staff will go out and collect that carcass. We're particularly interested in things like emaciated deer, because those could be an indication of chronic wasting disease. Raptor species that might be afflicted with highly pathogenic avian influenza, or if there are multiple dead animals, that's something we want to know about. The DEC staff will then submit those cases to either Del Mar or to Cornell, and we will take it from there. So on this slide, I have sort of a smattering of different diseases, some of which are zoonotic, which means that they can infect people. Some of those you are probably familiar with, such as rabies, but there's other diseases that we're constantly looking out for. Obviously, SARS-CoV-2 was a big one during the pandemic, and that did transmit into a number of white-tailed deer. Salmonella is one that we see frequently associated with bird feeders. We were fortunate not to find rabbit hemorrhagic disease in wild rabbits, but we did find it in domestic rabbits. Every year we monitor for West Nile virus. Snake fungal disease is an emerging disease over the past 2 decades, and then a variety of other viral and parasitic diseases. So we really are able to get cases from all over the state to examine a variety of species. Just in 2022, we saw 105 species of birds that resulted from 1,100 submissions. We had over 400 mammal specimens of 28 different species and 37 herpetofauna, so those are amphibians and reptiles, of 13 different species. And over the course of a year, we monitor trends. So in 2022, we did see more of a spike in cases submitted in the spring and kind of a downturn in the summer, where usually the summer is our busiest time. And our goal is to identify why those animals may be impaired. And so we have a variety of pathologies. Those range from bacterial, fungal, nutritional, parasitic, viral. We do have some that we are just not capable of determining what was going on with that animal. So we had over 1,600 of these specimens submitted. And in addition, the lab at Del Mar does a number of forensic examinations, and then we also collect specimens for research projects. And in combination with the surveillance activities, we want to make sure that we're prepared to respond to disease outbreaks. So we have a field response plan when we have high-profile diseases, and we have a specific interagency plan to deal with chronic wasting disease. We work regularly with the Department of Agriculture and Markets, as well as the Department of Health, so that they're aware of the situation in wildlife, and we're keeping the lines of communication and coordination open. Our group also does a lot of teaching and training, because preparedness and response goes hand in hand with that. So we offer a variety of in-person and virtual trainings for students and professionals alike to make sure that people are safe in the course of their professional activities. And we have a large number of ongoing collaborative research projects, and these are just a few examples. We have been looking at fisher, which are a mustelid, looking at their reproductive health, their exposure to rodenticide, and one that we're doing right now is lead from scavenging carcasses. That's a collaboration with SUNY ESF. And combined with that, we have a multi-state study looking at toxic contaminants in waterfowl. We do a variety of work on amphibians through eDNA, which is where we can sample the water to look for species and also pathogens. And we just published a paper last year on the impact of lead poisoning on bald eagles and their recovery in the Northeast. And there's a PhD student currently working on moose health. For the public, we want to make sure that we have a strong effort towards communications and outreach, including active Twitter and Instagram accounts for DEC and the Cornell Wildlife Health Lab that you can follow. And we have a number of disease fact sheets that are pretty popular. And these PDFs can actually be printed from the Cornell website, which I'll show here. And we try to update this with interesting information on a regular basis. So please go and check that out. And then the last one that I'll mention is that we want to make sure that we're supporting policy. And so our program is constantly evolving to meet the needs of the state agency, and we're happy to raise the profile of wildlife health at every opportunity. So thank you very much for the time and attention today.
Well, thank you, Kristen. All right, so now is time for questions and answers. So if you have any questions for our panelists, please put them in the chat, and John will kind of moderate the chat here. So John, that's to you.
Yes, thanks, Steve. Just a note to everybody, since we're a little short on time, if you look at your chat window in the lower, right down at the bottom, there are 3 dots. You can click on those dots and it'll offer you the option of saving the chat, which will save the entire chat as a text file. There are a number of questions that were asked that were already answered in the chat, so I won't repeat those, but you can go to the chat. The question for Christy: can anyone speak to the slash wall methodology that Cornell developed to keep deer away?
Sure. So that was developed by some of my colleagues, Peter Smallage and others, and it's basically a method to take residual wood following a timber harvest and kind of pile it up in massive piles around the perimeter of the harvested stand. And they've had quite a bit of luck preventing deer from accessing those areas following a timber harvest. The only issue is that you need to have a stand that has enough wood that would be not marketable to be able to create those walls. And so maybe for really very small landowners too, and you'd need to have kind of a pretty heavy harvest. So it may not be appropriate for all landowners, but they have had some very good success.
Thank you. Two questions for Christy. First, do you have local training, or do you do local training for the AVID initiative? And secondly, is there a minimum number of sites you recommend within a given area?
Okay. Yes, as for trainings, first of all, we have resources on our website, and we're just developing some short instructional videos. We were just filming those yesterday. But I do quite a few workshops, and I'm willing to go just about anywhere in the state to do those. So if there's a level interest in a community, I'm happy to come out and do that for you. And then in terms of how many sites, if you have a property, a given property with forest, I would say 1 site per stand, and a stand is basically in a forest is basically where there's similar tree species composition. So kind of if you think of it as an agricultural field, it would be a cornfield versus a soybean field. Obviously, forests are a little more diverse and complex than that. But if you had a hemlock stand and then you had a northern hardwood stand, that might be an example. So on any given property, maybe 1 per stand. But region-wide or landscape-wide, I believe, and Jeremy, you can correct me if I'm wrong, I believe we're shooting for about 100 sites per wildlife management unit. Okay, Jeremy's nodding yes.
Yeah, just want to remind everybody that a lot of the speakers, especially Jeremy, posted a lot of very useful program and resource URLs in the chat, so you can go to those if you're interested in any of those programs. And I think that really covers most of the questions, Steve. The ones that weren't answered directly in the chat. All
right, good. Well, this ends the biodiversity sessions, and I'm going to be passing it on now to Ann Corman, and she's going to moderate the climate policy sessions that are going to be coming up. So Anne, why don't you take over? If I don't know if John, if you have to make her a co-host as well.
Hold on a second, I'll find her. Give me a second.
Yeah, Annie Corman.
Oh, looking for her. She may not have joined yet. I'm not sure.
No, she's on.
She's on. Okay, great. Thank you. Oh. When I see her. Oh, there. Ann Clark? No.
No, Annie
Ann
Corman.
Corman. Got it. Sorry. Thanks. Okay, she's now a co-host, or should be. Anyway. Make co-host. There we go. She is. Okay. Thanks a lot, Steve.
Sure.
Thanks, Steve. We have about 10 minutes before we're going to start. We're going to start at 11 o'clock, so we have a little break, but I'll take it over from here, and I'll wait for my presenters and the attendees to come in. Great session.
Thanks. I think it went well. No big technical glitches, which is good.
And
Yes, very good. And John, you are our technical assistant, is that correct?
Correct, yeah. I'll make all of your speakers co-hosts, and if you want, I can handle the the chat-based questions, or if you want to handle it a different way, that's fine too.
Okay. Someone had suggested, Kate Darfler had suggested turning off the chat during the presentations. Did you find so it wasn't distracting? Is that
actually it was very, very useful, because what happened was some people had very technical questions for some of the speakers, and they answered them in a chat, and therefore we didn't have to take up the whole group's time with those questions. But we just prefer, if you have any, refer the group to the chat for the more technical answers. Yeah.
Okay, great. And if you're here to help us monitor that's great. I didn't realize someone would be here. So when we get to after the presenters and we get to the Q&A, so did you want me to read the questions, or were you reading them?
I was reading them. You could. I think it's more streamlined if I do, but I'd be glad to either summarize them for you or just have you take a look at them. Again, what we experienced in the previous seminar and other ones also was that there are a lot of kind of conversational questions, like what's the URL for the particular program that you mentioned, and so on. And so those are those are best answered in the chat. And the other questions I can do, but it's up to you if you want to kind of moderate the Q&A. That's might even work better.
Okay, so you think it's better if I see if you don't mind doing the Q&A, because then I can keep track of where they are in their presentations to try and keep folks on time, because I think
I have no problem. I'd be glad to do that.
Okay, yeah, that'd be great. I see David West here. He's one of the presenters.
Okay.
Hey, David.
Okay, made him co-host. Hey, David.
Great.
Let's see who else is joining.
Kate has her hand up.
See, Raya, Bob, and Terry, right? Okay.
I'm going to just check and make sure the share does what I hope it will do. Looks good.
I see your notes on the side. There you go.
You see my notes?
Not anymore.